Photographic Atlas of Developmental Biology. Welcome to Developmental Biology. This atlas will help you explore the fascinating life of E1 embryos of metazoans, multicellular organisms. Developmental biology is the study of the formation of a new organism. Development includes growth, increase in size and mass, differentiation, specialization, metamorphosis, change in body form from larva to adult, tissue repair and regeneration, regrowth of lost structure. Developmental biology uses many disciplines to analyze the principles and mechanisms of development. It analyzes the anatomical structure of the embryo. This is classically referred to as embryology, function of embryonic structures, and the genetic and molecular controls of development. Although there is wide variation in how an embryo develops, most organisms go through similar stages of development during their life cycle, Fig.E 1.1. Since we will be examining changes in the embryo over time, we should consider the best means to refer to the passage of time. Although we measure Tin1e in hours and days, developmental biologists frequently refer to embryos passing through a series of developmental stages, Fig 1.1. The use of stages in the form of a staging series is highly beneficial, since many species embryos develop at different rates. See Appendix A, Staging Tables. Model organisms. Since it is unethical to experiment on human embryos, developmental biologists have studied a variety of model organisms that provide important insights into human development. Unfortunately, no single model organism has all the features needed to answer all developmental questions. Model organisms are usually small, readily available, low maintenance, easy to breed, have short gestation periods, are fecund, produce large numbers of offspring, easy to manipulate in the lab, and inexpensive to obtain and house. Most of the discoveries made in model organisms are relevant to human development. Developmental biologists who perform surgical manipulations to understand development, often work with large robust embryos of amphibians and chickens. Transparent embryos of certain mollusks, echinoderms, and nematodes are useful for cell lineage experiments in which the developmental fate of cells is analyzed. New tracking dyes and genetic markers have allowed analysis of cell fate in opaque embryos. Developmental geneticists prefer animal models that breed readily in the lab, rapidly mature, have a relatively uncomplicated genome, and are easy to mutate. These include the plant, Arabidopsis thaliana, fruit fly, Drosophila melanogaster, nematode, Sinorhabditis elegans, zebrafish, Brachydainio ririo, frog, Xenopus tropicolis, and mouse, Mus musculus. With the advent of developmental molecular genetics and evolutionary embryology, it is important to understand development in a wide variety of organisms, since a host of similar molecules regulate developmental processes across species. Lani-1 nodal organisms are dioecious, exist as separate sexes, and some are sexually dimorphic, sexes are distinguished externally. Many use aquatic environments, marine or freshwater, to support development of their delicate embryos. Embryos of terrestrial organisms may develop within the mother's body until birth, or they can develop external to the one another, such as within the amniote egg, example chicken egg, which carries its own food and water supply. One way to classify model organisms is to examine the formation of the early embryonic gut, which appears as a hollow tube, initially closed at one or both ends, depending on the species. The end that opens first, or is open first, develops into either the mouth area as in protostomes, or anal area as in deuterostomes. Additional differences between protostomes and deuterostomes refer to cleavage patterns and chylome formation. Of all the model organisms studied, certain ones have been particularly useful to teach embryology and developmental biology. In this chapter, we briefly examine some developmental model organisms commonly studied in the teaching laboratory, Table 1.1. The protostomes we will cover include members of the phyla Nemertea, Mollusca, Nematoda, and Arthropoda, and the deuterostomes include members of the phyla Echinodermata and Chordata. Nemertea. Members of the phylum Nemertea are commonly called ribbon worms due to their characteristic shape. Adults can be up to 122 centimeters, 4 feet, long and 16 millimeters, 5/8 inch, wide. They are also called proboscis worms because of the presence of a proboscis that is used in feeding. Nemerteans are bilaterally symmetrical, unsegmented worms. The milky ribbon worm, Cerebrachelus lacteus, belongs to the class Anopla since its mouth is located posterior to the brain, and the order Heteronemertea since it exhibits indirect development in which it goes through a larval stage, Fig 1.2. Cerebrachelus is a benthic marine organism that burrows in the mud or sand in sheltered bays and estuaries. Its range is from Maine to Florida and Texas. Its breeding season lasts from May through August on the northeastern U.S. coast. Cerebrachelus is dioecious and sexually dimorphic. Ripe females are dull, brownish red, and males are bright red. These colors fade outside of the breeding season. A large ripe female can produce up to 50 million oocytes in one breeding season. Cerebrachelus is a useful model because it produces oocytes with a large germinal vesicle, nucleus, that complete meiotic maturation following insemination, spermoocyte fusion. Table AL in Appendix A shows their development. Hemollusca include chitons, limpets, clams, mussels, scallops, oysters, snails, sea slugs, squids, octopi, and chambered nautili, Fig 1.3. They are dioecious, usually not sexually dimorphic, and breed seasonally. Of the 50,000 species, only certain ones have been adopted as useful. Limnea, Ilionassa, and Nassarius, limpet, patella, surf clam, spisula, and mussel, Mytilus. Their rapidly developing embryos are excellent models to study fertilization, cleavage, gastrulation, and larval stages, Table A.2. Fig. 1.4 shows a mollusk larva called a trochophore. Depending on the species, the trochophore will either metamorphose directly into a young adult, or develop into a second larval stage, the veliger, which later one metamorphoses into an adult. Nematoda. Nematodes that are useful developmental models include the free-living nematode, Sinorhabditis elegans, and the parasitic roundworm, Ascaris, Parascaris. While C. elegans is soil-dwelling, adult Ascaris lives in the intestines of its host including pigs, Ascaris suum, horses, Parascaris equorum, and humans, Ascaris lumbricoides, Fig 1.5. C. elegans is only 1 nm long, making it an excellent model to rear in the lab. Adults exist as males and self-fertilizing hermaphrodites, and live about 2 to 3 weeks. Although it takes only 3 days to develop from fertilized oocyte to mature adult, C. elegans develops a full set of differentiated tissues, Table A.3. C. elegans is transparent at all stages during its life cycle, and thus the fate of every single cell is known. Its entire genome of 5 autosomes and 1 X chromosome has been sequenced, and subjected to intense genetic analysis. Ascaris, which is dioecious and sexually dimorphic, ranges in length from 15 to 40 centimeters with males smaller than females. Its diploid chromosome number is 4, 2n. Although opaque, Ascaris is a useful teaching model because sections of the female reproductive tract readily show all stages of meiosis, fertilization, and early cleavage. However, it is a challenging research model because it is difficult to maintain in the laboratory, is parasitic in humans, and highly allergenic. Arthropoda. He major arthropod developmental model is the fruit fly, Drosophila melanogaster, which belongs to the class Insecta, Fig 1.6. Drosophila are dioecious and sexually dimorphic. Adults live about 2 to 4 weeks, and are 3 mm long. Fertilization is internal and females lay up to 100 fertilized oocytes in one day. An embryo develops into a first instar larva in one day, molts twice to form a third instar larva which pupates and reaches sexual maturity in 6 days. Table A.4. Although the embryos are opaque, the larval stages have been useful to study behavior of polytene cron 1 osomes during development, Fig. 1.7. Drosophila have 3 autosomes plus an X and a Y chromosome, and its genome is sequenced. Many of the genes involved in formation of the body plan have been discovered in Drosophila. It is a powerful model organism for developmental geneticists and evolutionary biologists. The lubber grasshopper, Romalia, is a useful teaching model because sections of the testis demonstrate stages of meiosis, Fig. 1.8. Echinodermata. Echinoidea. The sea urchin is a classic embryological specimen and an excellent model to study fertilization and early development, Table A.5. Adults produce millions of gametes, oocytes, spermatozoa, in one season that are easily fertilized in the laboratory. When fertilized, the transparent ova, eggs, develop synchronously, making them a powerful model to study fertilization and embryonic development. Sea urchins are benthic marine invertebrates. They are dioecious, but not sexually dimorphic. Rare hermaphrodites exist. Sea urchins are seasonal breeders in that they spawn only during specific tin-1es of the year. For example, Lidocynus pictus. Fig. 1.9, produces viable gametes from late spring to early fall, L. variegatus from fall to spring, Arbatia punctulata from June until August, and Strongylocentrotus purpuratus from December to 11I in the United States. Asteroidea. Starfish, Asterias, are dioecious, and not sexually dimorphic. Fig. 1.10. They are benthic marine organisms that seasonally spawn. Asterias spawn in May through early July in the United States. Their transparent oocytes undergo stages of meiosis when shed from the ovary, thus they are important models to study meiotic maturation, fertilization, and embryonic development. Chordata. 1/J members of the phylum, Chordata, which include LV. Tunicates, and vertebrates, are characterized by the presence of a dorsal hollow nerve cord, notochord, pharyngeal slits, and a muscular postanal tail at some time during their embryonic and/or adult life. An additional feature is the presence of a subpharyngeal organ, endostyle or thyroid gland, that binds iodine. These are called chordate characteristics. Fig. 1.11. Cephalochordata. The cephalochordate developmental model is Branchiostoma, which is also called lancelet, means little spear, and Amphioxus, means sharp at both ends. There are 29 known species with a 6 cm maximal length. They inhabit warm, shallow subtropical and tropical marine environments around the world. They live in burrows made of sand, while filter feeding on is an omnivore that eats by scraping surfaces with its mouth, seen in B on phytoplankton. They are dioecious and not sexually the ventral side. Dimorphic. Mature males have creamy white testes. And mature females have lemon yellow ovaries that can produce thousands of oocytes 140 μm in diameter, Figs 1.12, 1.13. They spawn while swimming and then return to their burrows. They can spawn several times in one season. They are useful to study meiosis, fertilization, and embryonic development. Table A.6 shows their development. Osteichthyes adenopterygii. 2 excellent species of tropical freshwater teleost fish have been used to study the cellular, molecular, and genetic mechanisms of vertebrate development. These include the zebrafish, Brachydanio, Danurerio, and Japanese killifish Medaka, Oryzias latipes, Figs 1.14, 1.15. They are hardy animals that are easy to care for in the laboratory. They range from 2 to 5 centimeters in length, are dioecious, and sexually dimorphic. During spawning, zebrafish females can release up to 100 or more oocytes daily. Oocytes and embryos are transparent, enabling visualization of cell movements and developing internal organs, Fig 1.15. Embryos develop rapidly, hatching in 3 days at 28.5°C and becoming sexually mature in about 3 months. Zebrafish, which live about 5 years, have 50 diploid chromosomes that allow genetic manipulation. Staging series have been developed, Table A.7. Amphibia. Useful developmental models include frogs, anurans, tailless amphibians, and salamanders, urodels. Robust embryos that develop externally and are amenable to microsurgery. Amphibians are seasonal breeders. While the population of the wild leopard frog, Rana pipiens, is on a serious decline in the U.S., developmental biologists have turned to a frog model from Africa called Xenopus laevis, African clawed frog, or toad. Xenopus are dioecious and sexually dimorphic with adult females larger than males, Fig 1.16. It takes 1 to 3 years to reach sexual maturity, and they have a lifespan of 6 or more years. Staging series have been developed, Table A.8. Because X. laevis is tetraploid, with 36 chromosomes, making genetic studies difficult, developmental biologists have turned to the smaller frog, Xenopus tropicolis, which reaches sexual maturity in less than 5 months. X. tropicolis is diploid with 10 chromosome pairs, making genetic manipulation feasible. Frog embryos develop into free-swimming, self-feeding tadpoles which later metamorphose. During metamorphosis, the gills and tail are resorbed, and limbs develop. Aves. The most widely used classical developmental model is the domestic chicken, Gallus gallus, Gallus domesticus, Fig 1.17. Quail embryos have also been used. Birds are dioecious and chickens are sexually dimorphic. Avian embryos are large and robust enough to survive surgical manipulation. Because the environment, temperature, can control the rate of development, in 1951 Hamburger and Hamilton staged chick embryos based on their morphological features rather than incubation times, Table A.9. In 1976, Ayal and Kochov created a staging series for the very earliest stages of chick development. If incubated at 38°C, chick embryos hatch 19 to 21 days after laying. Their pattern of development is similar to mammals and thus are an important one no doubt to understand development. Mammalia. The most widely studied mammalian model is the mouse, Mus musculus, Fig 1.18. While mice exhibit internal fertilization, oocytes can be fertilized in vitro, in the lab, and develop asynchronously for about a week, after which they need the uterine environment for survival. Murine oocytes and early embryos are transparent, making them amenable to light microscopy. Mice live up to 3 years and reach sexual maturity in 4 weeks. Their gestation period is 19 to 21 days and females bear 1 to 10 pups, Table A.10. Mice have 19 autosomes and 2 sex chromosomes, X, Y, and the entire genome is sequenced. Transgenic mice, mouse knockouts and knockdowns, and genetically engineered mice have provided a powerful means to study development and human disease. Rat embryos have also been studied, but to a lesser extent, Table A.10. Because rodent embryos and fetuses are difficult to work with and undergo embryonic turning, which one makes their anatomy challenging to interpret, the most widely used model to teach one mammalian embryology is the Do-1 nestic pig, Sus scrofa, Fig 1.19. Table A.11 shows embryonic island of the pig. In this atlas, the monkey has been used to demonstrate histology of gonads and reproductive structures. The fascinating world of embryos is too small to see details with the unaided eye, therefore light microscopes are extensively used. Embryos are visible with the compound light microscope or the stereo, dissecting, light microscope, depending on the size, thickness, and transparency of the specimen. There are many different styles of both stereo and compound microscopes, however, they share common features which // single quote will be described here briefly. Knowing when and how to use which type of microscope greatly aids the observer. Also, knowing how to handle and interpret the slides makes the study of embryos informative and rewarding. Operating the stereo light microscope. Stereo microscopes are typically used to observe surface details of non-transparent embryos, or to examine relatively large, transparent samples at low magnification, Fig 2.1. For optimal viewing with the stereo microscope, the following Co-1 endpoints need adjustment each session the microscope is in use. 1. Interocular distance or interpupillary distance, distance between eyepieces. 2. Focus of each eyepiece. 3. Lighting, whether from above or below the specimen. 4. Focus of the specimen. And 5. Zoom, magnification, of the specimen. Fig 2.2 shows the striking effects of different lighting conditions on the appearance of an embryo. Samples typically examined with the stereo microscope include unmounted living and preserved specimens, and embryos mounted on slides, whole mounts. Sectioning. See internal details of an embryo. The embryo is fixed and cut into thin SC sections using a device called a microtome. Several section planes are used, Fig 2.3. Most sections are 7 to 15 μm thick and start at one end of the embryo, anterior or dorsal, and move to the other end, posterior or ventral. The delicate sections are carefully mounted on a microscope slide in the order in which they were cut. They are then stained usually with hematoxylin and eosin, so that one can visualize the otherwise transparent tissues. Hematoxylin is a basic stain, making acidic components of the embryo, such as DNA, a bluish purple. Eosin is an acidic stain, making basic cellular components, such as cytoplasm and smooth endoplasmic reticulum, pink. After staining, the sections are covered with a cover slip to prevent damage. Cover slips can be round, square, or rectangular, but are always extremely thin and fragile, Fig 2.4. The convention for orienting sections on the slide is shown in Fig 2.5. Sections are mounted in horizontal rows, to be viewed from left to right, like the text of a page. If there are too many sections for one slide, they may be mounted on two or more slides. A series of slides that have every single section of the E1 embryo is called a set of serial sections. If an embryo is large, hundreds of sections may be created and not a single one is lost. It takes great skill to create serial sections. A transverse serially sectioned 21-hour chick embryo fits on only one slide, but a 96-hour chick embryo may fit on 6 slides. For embryos on multiple slides, predicting which slide contains the structure of interest greatly improves efficiency of study. Sections provide only a two-dimensional view of the structures within an embryo. To get the most information from a serial section series, one must mentally three-dimensionally reconstruct the relations of the different structures to each other along the length of the embryo. This skill takes time, and is very rewarding, for it enables a true understanding of the complex and dynamic structure of an embryo. Handling slides. Removing slides from the box. Slides are stored upright in boxes or flat in folders, Fig 2.6. Before opening the storage container, make sure it is not upside down and that the slides are upright or in a stable condition. Some boxes need to be squeezed in the middle of the sides before the lid will come off, others have a hinge which needs to be undone. Handle the slide only at the areas where the cover slip is absent. When in doubt, only grasp the edges of the slide or the area with the label, Figs 2.7, 2.8. Always remember where the slide came from and return it to the same spot. Usually information is provided on the lid as to the location of the slides. This is especially important for serially sectioned embryos. When returning thick specimens to the box, make sure other slides in the container will not bump them. Never place anything heavy on the slides, slide box, or folder. This can crush the delicate embryos. Reading the slide label. Each slide has a label on which is found the following: manufacturer, catalog number, organism, embryonic stage, plane of section, or if whole mount, series number if serially sectioned, slide number if in a series, and any special information about the specimen or staining, Figs 2.4, 2.5, and 2.8. Reading the label is especially important for serial section series, so that it is understood which slide of the series has been chosen. The last slide in the serially sectioned embryo usually has an X after it, Fig 2.5. For example, if a 24-hour chick serial transverse series is on only one slide, that slide is marked, slide LX. If the class has 20 serially sectioned 10 mm pig embryos, and Series 3 contains 12 slides, the 12th slide of Series 3 is marked SIR 3SL12X. The 7th slide of the series is marked SIR 3SL7. The second slide of Series 9 is marked SIR 9SL2. Preventing damage. Slides can last over 50 years if taken care of properly. Some embryonic samples are no longer commercially available and thus are irreplaceable. A few simple steps can prevent damage to the slides. Table 2.1. When one slide in a serial section series is damaged, the whole set is ruined. Instructors expect the slides to be in the same condition at the end of the course as when they were first handed to students. Inspect the slides carefully when first received, and report any damages immediately to the instructor. Operating the compound light microscope or comfortable viewing with the compound flight microscope, several components need to be adjusted at the beginning of each viewing session since the previous users have adjusted the one microscope differently for their needs, Fig 2.9. The components that need adjustment include 1. Interocular distance, 2. Individual focus of the eyepieces, if a binocular microscope, 3. Lighting, make it not too bright, 4. Iris diaphragm, controls contrast, 5. Focus control knobs, focus up not down, 6. Choice of objective lens, and 7. Position of embryo or section on the stage, use the stage control knobs and vernier scale if present. It is critical that the optical surfaces of the microscope and viewing area of the slide are free of fingerprints which smear the eye, making viewing unnecessarily difficult. If cleaning optical surfaces is necessary, only use lens paper for the job, never paper towels, tissues, or Kim wipes which permanently scratch and ruin glass surfaces. Touch surfaces lightly, never rub. Lighting. Before turning on the microscope, adjust the rheostat knob that controls the lighting so that it is the dimmest possible. Turn on, press, the on/off switch, and slowly adjust the rheostat to a comfortable level about midway. Viewing samples with bright light causes loss of detail, and can cause a headache. Living and unstained samples can be bleached out by bright light, making them difficult to find. To achieve the highest possible contrast during viewing, close the iris diaphragm to make features of the sample visible, Fig 2.10. Oculars. Adjust the interocular distance on the binocular microscope to a comfortable level for your eyes, Fig 2.11. Use the interocular scale to make a note of this distance. Always set the microscope to this distance during viewing. If glasses are worn, keep them on during viewing, but be careful not to press them against the oculars so that the lenses don't get scratched. Many oculars have rubber cups to reduce stray light at the eyepieces, while allowing the viewer's eyes to be at a comfortable distance from the eyepieces. For those with eyeglasses, the rubber cups can be peeled back to allow for proper viewing distance and to prevent lens damage by the oculars. Since vision in each of the viewer's eyes is not identical, usually one ocular is fixed while the other is adjustable. This allows individual focus of the eyepieces. With the eye above the adjustable ocular closed, focus the sample using the fixed ocular, see section on focusing. Then open the eye above the adjustable ocular, keeping the other eye closed. Without changing the focus control knobs near the stage, rotate the collar on the adjustable ocular, to get the specimen in crisp focus. This step will prevent eye strain. It is important for users of monocular microscopes to keep both eyes open during viewing, and to mentally ignore the visual information from the open eye not looking through the microscope. To prevent fatigue, viewing can be switched between eyes. Changing magnification. Typically 4 objective lenses are used on the compound light microscope: 4x, low power, 10x, medium power, 40x, high dry power, and 100x oil immersion, high power, as seen in Figs 2.9, 2.10, and 2.12. The type of lens is marked on its barrel with a number and a colored line. Star, T each viewing session at low power. Objective lenses on the microscope may not be parfocal. When one objective lens is in focus, the other objective lenses on the microscope are also in focus when in use. Therefore, after changing objectives, view the new objective lens barrel from the side. Periodically during refocusing to ensure that the embryo will not be crushed. To change objectives, rotate the nosepiece rather than pushing the objective lens barrel. The apparent magnification of a specimen is the objective lens power multiplied by that of the oculars. When viewing an embryo with a typical 1-ox ocular and 1-ox objective lens, it will appear 100x bigger than with the unaided eye. Focusing. Know what the sample is expected to look like by consulting the proper chapter of this atlas. First, view the slide with the unaided eye. Is the area of interest of the sample visible? If so, position that area under the lowest power objective lens. Is the height of the sample and cover slip thick as in Fig 2.4a? If so, keep in mind the working distance between the bottom of the objective lens and top of the specimen, of the objective lens. With increasing magnification, the working distance is greatly reduced, Fig 2.12, and the danger of accidents increases. To view samples, start with the lowest possible magnification and make sure the bottom of the objective lens is far from the top of the cover slip. Viewing of serial sections becomes efficient when focusing with one hand and moving the slide with the other hand. If the sample is not visible with the unaided eye or stereo microscope, start at 10x. With the compound light microscope and first focus on the left or right edge of the cover slip. Adjust the iris diaphragm for maximal contrast. Then slowly move the center of the cover slip into view while looking through the eyepieces. Adjust the focus when any blurry structures appear to move across the field of view. If necessary, ask the instructor for help. Tracing. An important activity when viewing serial sections is to trace structures in the series, Fig 2.13. For example, when viewing a serial transverse series of a 4 mm frog embryo, stage 18, one can trace sections posteriorly and watch the neural tube expand to form optic vesicles in the brain area and narrow again to form the spinal cord. In the same series, the large foregut can be traced as it narrows posteriorly to form the midgut and hindgut. With large serial section series, viewing slides first with the unaided eye and/or the stereo microscope for orientation, help in the choice of section to view on the compound light microscope at low magnification. Use caution when tracing structures in serial sections, since the rapid motion during viewing can cause motion sickness. Interpreting slides. Two embryos at the same embryonic stage are exactly alike. In sectioned material, the plane of section may be different than that shown in this atlas. Refer to the whole mounts often to get the big picture. Understanding the general features of what to expect by familiarizing yourself with this atlas before studying the slides is one of the best ways to prepare in advance of slide study. It is helpful to view multiple specimens of the same stage to gain the best understanding of embryonic structure. When first examining the embryo, always try to identify its axes: anterior, posterior, dorsal, and ventral. This is especially important with sectioned embryos. Orienting the slide on the microscope so that it matches the view in this atlas helps. This may entail rotating the slide on the microscope stage 180 degrees. However, never turn a slide upside down so that the cover slip faces downward. The later stage embryos have several tubes within a tubular body. For example, the central nervous system, gut, and blood vessels appear as hollow tubes with dilations and constrictions in the embryo. Other structures such as the rod-like notochord appear as a solid circle or oval in transverse sections. Fig 2.14 shows how a tube can appear. In sectioned specimens depending on the plane of section. Blood vessels, which should be hollow, may look solid if full of blood cells. This happens if the embryo was lying on its side for a prolonged period during fixation. When looking for paired blood vessels, the set of vessels on one side may appear hollow, while those on the other side appear solid. Chick embryos, 48 hours and older, and mammalian embryos curl upon themselves during development, forming a question mark or C shape. In transverse section, these embryos appear to have two brains when in reality the curled head has been cut twice. Frequently consulting images of the whole mount and correlating these with sectioned material helps prevent confusion. Don't hesitate to ask the instructor for help. Our analysis of how metazoans develop begins by considering the organ systems responsible for producing gametes, sex cells. The reproductive organs or gonad, ovary, testis, produce gametes, ovum, spermatozoon, through gamerogeny, meiosis. Meiosis is a critical event that impacts the life of every multicellular organism that reproduces sexually. Mistakes during meiosis can lead to a variety of abnormalities ranging from mild to severe, and can cause death of the embryo. Meiosis is a two-step process whereby two cell divisions give rise to gametes, Fig 3.1. The normal diploid number of chromosomes is halved during meiosis and later restored during fertilization. Gametogenesis is called Spermatogenesis in males, and Oogenesis in females. Common features of these processes will be discussed here. Spermatogenesis is easier to understand, and its specific details are covered in this chapter, whereas Chapter 4 specifically addresses Oogenesis. The generation of mature gametes involves three stages: primordial development, pre-gamerogenesis, and gametogenesis. Spermatogenesis itself consists of two major phases: meiosis, which produces a haploid male gamete, and spermiogenesis, which modifies the male gamete, streamlining its structure so that it can successfully fertilize the female gamete. Primordial DEVE.L, O, PME, N, T, A _ N dot comma _ DPR, e.g. A, M, ET, O, G _ E _ N, ESIJOS ___. Before studying meiosis, it is important to understand where progenitors of reproductive cells called primordial germ cells, PGCs, come from. PGCs in females are called oogonia and spermatogonia in males. In insects, PGCs originate from pole cells located in the embryo's posterior. During pre-gametogenesis, pole cells undergo mitosis and migrate to the posterior midgut. Eventually PGCs migrate to the developing gonads where they become surrounded by mesoderm cells, embryonic germ layer. Vertebrate PGCs form and enter the gonads differently. Prior to gastrulation, reptilian, avian, and mammalian embryos are two-layered. They have an upper, dorsal, epiblast layer and a lower, ventral, hypoblast layer. During mammalian primordial development, PGCs are produced in the posterior region of the epiblast. During pre-gametogenesis, PGCs migrate to the endoderm, embryonic germ layer. After residing there for a few days, they migrate to the genital ridges, developing gonads derived from mesoderm, where they increase in number by mitosis. After mitosis ends, they enter a growth stage. At that time they are referred to as primary gonocytes, primary oocytes and primary spermatocytes. Like insects and mammals, amphibian PGCs migrate from the endoderm to the genital ridges. Unlike these PGCs, Avian and reptilian PGCs migrate from the epiblast to the germinal crescent, an area in the anterior hypoblast, and then to the genital ridges through the bloodstream. Meiosis. Upon appropriate cues, primary gonocytes enter first meiosis, Fig 3.1. Meiotic prophase consists of 5 individual stages: leptotene, zygotene, pachytene, diplocny, and diakinese. Homologous chromosomes, chromosomes with different versions of the same genes, condense during leptotene, pair during zygotene, exchange DNA, crossover or recombine, during pachytene, separate during diplotene, and move apart to the nuclear periphery during diakinesis. At pachytene, each pair of homologous chromosomes is a bivalent of 2 chromosomes and a tetrad of 4 chromatids. They align on the meiotic spindle during metaphase I and segregate to opposite spindle poles during anaphase I. Homologous chromosomes are equally distributed to 2 daughter cells during anaphase I. At the end of first meiosis, the daughter cells, called secondary gonocytes, secondary oocytes and secondary spermatocytes, are haploid with each chromosome containing 2 chromatids. The secondary gonocytes then enter meiosis II upon receiving appropriate cues. Sister chromatids, which were held together at the centromere, are separated and distributed equally to the 2 haploid daughter cells called gonadids, ootids, oocytes, spermatids. Gonadids may then undergo further cellular modifications before being considered mature gametes, ova, spermatozoa, capable of fertilization. Spermiogenesis. Although haploid, spermatids are not capable of fertilizing a female gamete. For successful fertilization, a mature male gamete must reach the ovum, penetrate and activate it, and contribute the paternal genome to it. The structures and organelles that give the male gamete these capabilities develop during spermiogenesis. In many metazoans, several components of the spermatid are altered during spermiogenesis to streamline the sperm. 1. Chromatin changes protein composition to arginine-rich proteins, allowing the chromatin to become highly compact. 2. The nucleus changes shape, often elongating. 3. An acrosomal cap forms in the head of the sperm. The acrosome is a highly specialized lysosome that allows the spermatozoon to penetrate the extracellular matrix of the female gamete. 4. In most species, a flagellum grows out from one of the centrioles located near the nuclear membrane. The flagellum provides a means of motility, allowing the sperm to reach the female gamete. 5. Mitochondria concentrate around the axoneme of the flagellum, providing a source of energy for flagellomotor proteins. 6. Excess cytoplasm is pinched off and discarded in the form of N-sidual HOTIC. Once these cellular modifications are completed, the mature male gamete is called a spermatozoon, Fig 3.2. Anatomy of the testis. During early fetal life of mammals, the testes first develop in the abdominal cavity in close association with the embryonic kidneys, mesonephros. As the testes grow, they migrate down through the inguinal canal and abdominal wall, and into the scrotum. During migration, the testes become enclosed in a flask-like double-layered sac of the abdominal peritoneum called the tunic. 1 Yagin.3. The cavity formed by the tunica vaginalis contains the testis and epididymis, storage area for sperm, and is continuous with the abdominal cavity. Exiting the testis is the PCRM.Uic cord, which contains the vas deferens, blood vessels, and nerves. When the migration pattern of the testis is complete, the testes are arranged as shown in Fig 3.3. Testes of mammals with low body temperatures, whales, monotrams, elephants, remain in the abdominal ca/iry. Testes vary in size, shape, and weight, Fig 3.4. In birds and mammals, the testes are paired, ovoid structures of equal size, but there may be a single or multiple testes in other organisms. The adult human testis is smooth and 4 to 5 cm long by 3 cm wide, weighing 15 to 19 grams. In contrast, A single testis of the adult Indian elephant weighs 2 kilograms. In echinoderms, insects, and fish, the testes are lobular instead of smooth. Each adult mammalian testis is located within the scrotum, a sac-like fold of the body wall that suspends the testes away from the body, keeping the testes cool, a requirement of spermatogenesis. Temperature regulation of the testes is accomplished by subcutaneous muscles of the scrotum called the cremaster/mu/CLC. Each cremaster muscle contracts when cold and relaxes when warm, thus changing exposure of the testes to the warmth of the body. Histology of the testis. Spermatogenesis is seasonal in many organisms and under hormonal control. Men produce sperm continuously, up to 150 million sperm per mil semen in a 2 to 6 ml ejaculate. To produce the high numbers of sperm, the testis needs a large surface area. Mammalian sperm are produced in seminiferous tubules, which are convoluted hollow tubes about 30 to 70 centimeters long and up to 250 m wide, Figs 3.5, 3.6. A thick fibrous covering called the tunica albuginea surrounds the testis. Extensions of the tunica albuginea deeper into the testis form septa and lobules. Up to 4 seminiferous tubules are packed into a lobule, and there are about 250 lobules in the adult human testis. The septa converge at the diasternum, which is a connective tissue framework that supports blood and lymph vessels, and afferent ducts as they leave the testis. Mature sperm are stored for a few weeks in the epididymis, until their release into the vas DCFCRCNS. The epididymis is a single convoluted tubule about 6 m in length, one on each side of the body. It lies next to, but is not a component of the testis. In fish, amphibians, and some reptiles, the epididymis is either rudimentary or absent, sperm drain directly into the vas deferens. The pathway of sperm release from the human testis is seminiferous tubule, produce mature sperm that are released MTO the lumen, hollow center. Straight tubule, tubuli recti drain seminiferous tubules. RCTC testis, network of tubules in mediastinum that drain the straight tubules. Efferent ducts, ductolike phalanx, transport sperm in rete testis into epididymal head. Caput, head, of epididymis, receives immotile sperm from efferent ducts and stores them. Corpus, body, of epididymis, slowly drains the caput and stores increasingly motile sperm. Cauda, tail, of epididymis, slowly drains the epididymal body, and is the main storage site of sperm, -50% motile, until they are ejaculated. Vast phragm, ductus deferens, a -30 cm muscular duct, one on each side of the body, which carries sperm from the cauda epididymis upward through the inguinal canal, into the abdomen, behind, dorsal to, the urinary bladder, and down to the level of the prostate gland, where it joins with the duct from the seminal vesicle to form the ejaculatory duct. The proximal end, part closest to the testis, is highly convoluted. The seminal vesicles contribute fluid to the sperm suspension. Ejaculatory ducts, embedded in the prostate, the right and left ejaculatory ducts drain the respective vas deferens and seminal vesicle, and carry sperm through the prostate to the urethra. Additional secretions from the prostate gland are added to the sperm suspension. Urethra, a single 15 to 20 centimeters long duct which is embedded in the prostate, upper portion, and penis, lower portion. It drains the 2 ejaculatory ducts and bulbourethral glands which contribute fluid to semen. When not involved in reproduction, the urethra also drains the urinary bladder. The urethra is the last duct sperm pass through before leaving the body. Dash, comma dash dash comma underscore 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 comma underscore dot comma underscore comma. Spermatogenesis in model organisms. Testicular histology appears different in diverse species. However, despite the variation, There are common themes in spermatogenesis. One theme is that spermatogenesis occurs unidirectionally as a wave in the testis, beginning with spermatogonia and ending with mature sperm. How this is achieved varies amongst species. This section demonstrates spermatogenesis in a variety of model organisms. Starfish. During the reproductive season, starfish testes are enlarged and lobulated, Figs 3.7 to 3.9. One to several testes are located in each arm. Spermatogonia are found at the periphery of each lobule and undergo meiosis, moving progressively inward as they mature. Mature sperm are released into the lumen of each lobule where they are stored until needed. Grasshopper, Romalia. Testes are paired and possess several lobes, also called follicles or sperm tubes, lined with a basement membrane and epithelial sheath, Figs 3.10 to 3.15. A peritoneal sheath of connective tissue surrounds the group of testicular lobes. The proximal, pointed, end of each lobe is connected to the vas deferens, primary exit duct of each testis, by a short tube, the vas efferens. Both vas deferens drain into a common ejaculatory duct. At the distal, apical or blunt, end of the testicular lobe, which is farthest from the vas efferens, spermatogonia undergo mitosis and are encysted. All the cells in each cyst are clones, and undergo meiosis synchronously. Spermatogenesis occurs in a wave down each lobe. In the middle of the lobe, the encysted cells undergo first and second meiosis, while spermiogenesis occurs at the proximal end of the lobe. New cysts at the distal end of the lobe push the more meiotically advanced, older cysts proximally. Eventually sperm break out of their cysts, enter the vas efferens, and drain into the vas deferens where they are stored until needed, Fig 3.16. Male grasshoppers have one X chromosome and no other sex chromosome, XO, whereas females have two X chromosomes, XX. During first meiosis in males, the X chromosome is condensed and readily visible in the nucleus. L.Ansia, Amphioxus, Branchiostoma. Testes are hollow and fill with sperm during the reproductive season, Figs 3.17, 3.18. They take up a large volume of the body cavity. The ripe testes burst, releasing mature sperm into the atrium, which is a cavity that surrounds the pharynx, extends posteriorly to the hindgut, and houses the paired gonads. The atrium opens to the outside via the atriopery, which is anterior to the anus. Water flowing posteriorly in the atrium carries sperm to the outside via the atriopery. After sperm release, the testes reseal and spermatogenesis can begin again. Fro. Located on the ventral side of the kidneys are the paired, oval testes of the frog, Fig 3.19. Spermatogenesis is seasonal and occurs in cysts located in seminiferous tubules, Figs 3.19 to 3.27. Immature cells are in the periphery of the seminiferous tubules, and mature sperm are released into its lumen. Germ cells are supported by columnar-shaped, Sertoli cells, which are also called nurse cells. Sertoli cells provide physical support and supply nutrients to developing sperm. Mature sperm exit seminiferous tubules through collecting tubules. Interstitial cells, L.C. dig cell, are found outside the cysts, and separate seminiferous tubules from each other. Interstitial tissue, supporting connective tissue, is continuous with the tunica albuginea, which is the outer connective tissue covering of the testis. A double fold of peritoneum called the mesorchium covers each testis and attaches it to the kidney. Between folds of mesorchium are ducts from the testes, called the vasic fertinia, which drain sperm from collecting tubules. Bird. Avian testes are paired and located mid-dorsally within the abdomen on the ventral side of the kidneys, Fig 3.28. Each testis is covered by a tunica albuginea. Avian spermatogenesis occurs at 40 to 41 degrees Celsius, unlike mammals, 35 degrees Celsius or lower. Spermatogenesis is seasonal and takes place within seminiferous tubules, Figs 3.28 to 3.31. Immature sperm. and certola cells are located at the periphery of the uraniferous tubules, whereas mature sperm are released into the central lumen. Certola cells provide physical and nutritive support for developing sperm. Sperm are stored in the epididymis Fig 3.32, and released into the vas deferens. Deferent duct, to the female reproductive tract. Mammal. Mammalian testes are packed with seminiferous tubules, Figs 3.33 to 3.59. Each seminiferous tubule is surrounded by a basal lamina, thin basement membrane of connective tissue, and then the tunia propria, thin layer of connective tissue. Contractile myoid muscle-like cells, lie outside the tunica propria in non-human testes. Between the seminiferous tubules are blood vessels and interstitial cells, Leydig cells, which respond to luteinizing hormone by secreting testosterone. The wall of each seminiferous tubule is the germinal epithelium, which contains certola cells, sustenacular cells, and several layers of spermatogenic cells, Fig 3.6. Certola cells function to nourish developing sperm, provide physical support, and respond to follicle-stimulating hormone by secreting an androgen-binding protein. Spermatogenesis occurs unidirectionally as a wave in the germinal epithelium, with the most immature cells located in the periphery of the tubule, and mature sperm are in the innermost layer where they are released into the lumen. Mammalian spermatogenesis begins with type A spermatogonia, which line the inner surface of the basal lamina, Fig 3.60. Type A spermatogonia divide mitotically several times to produce more of themselves, and type B spermatogonia. Each type B spermatogonium produces two diploid primary spermatocytes, each of which divides during first meiosis to produce two haploid secondary spermatocytes. Each secondary spermatocyte divides by second meiosis to produce two haploid spermatids. Thus, through two meiotic divisions, one spermatogonium gives rise to a total of 4 spermatids. Once spermatids are formed, they undergo spermiogenesis to become mature sperm. This takes 24 days in humans. It takes about 64 days to produce a mature human spermatozoon from a spermatogonium. Spermatozoa drain into the straight tubules, then the reedy testis, and leave the testis through efferent tucrals into the epididymis, where they are stored, Figs 3.61 to 3.65. Sperm exit the body through the vas deferens and urethra, Figs 3.66 to 3.67. Fluid from the seminal vesicle and prostate gland is added to the sperm suspension, Figs 3.68 to 3.70. Sperm from different species vary in shape and size, Fig 3.71.